we've talked about Mendelian genetics, we've talked about chromosomes, right? And this whole time we've kind of been glossing over what the DNA has been doing. We've talked about all these different things that are kind of the result of what the DNA is doing. Let's knock the lights down, I guess, so we can actually see the stuff on here. And so chapter 14, we're actually going to look at the structure of DNA and how DNA is getting copied. All right, so chapter 14. Okay, good. So real briefly, we're going to kind of give a little bit of background. I'm going to go over this. I've kind of trimmed this lecture down a little bit just because it had a lot of extra stuff in it that I don't think is critical for us. But we're going to just talk about the nature of the genetic material, right? What actually is it? How is it coding information? What does it look like? All of that good stuff. So, right, we mentioned this, early 20th century, people found all of Mendel's stuff, right? And then we talked about in chapter 13, where we figured out that these traits that Mendel was observing are coded for, right? The information are on the chromosomes. But if you remember all the way back to like chapter 3, maybe, when we talked about DNA, right? Chromosomes, an actual condensed chromosome, is made up of DNA and protein, right? It's DNA wrapped around those little histones, all kind of coiled up and packed together, right? And so initially people were looking at this. So the chromosomes are carrying the traits, right? We figured that out. We looked at flies. We did all this stuff. The chromosomes have the information in them. But is it in the DNA or is it in the proteins? And initially, people were assuming it was in the protein. And for logical reasons, right? DNA just has those 4 little nucleotide bases, right? A's, T's, and G's. How much information can you make out of 4 little bases versus proteins, which were made of, right, are linked up chains of 20 different amino acids, which is kind of close to our own alphabet. So like logically, people were thinking that the protein was carrying the information and that the nucleotides were just kind of this rope that was holding them all together. Of course, we now know that that's incorrect, but move on. There's a whole lot of, like, if you want to kind of get a little more, there's a whole list of experiments and things in the chapter, but I don't think we really need to spend a lot of time on it. If you're curious, it's in there. So DNA structure. So the DNA is what's holding the information, these 4 little nucleotides. Fair, fine. And so what does it actually look like, right? Like, what does the molecule actually look like? And we, at this point, right, it gets drawn on a lot of stuff. You know, like DNA is a double helix, but how did we actually get there? Credit for DNA structure is given to Watson and Crick, but there's kind of a long path to get there. So DNA itself, like the recognition of the existence of the molecule DNA, was discovered by Friedrich Miescher. You don't really need to know Miescher's name, but he's the guy who did it back in the 1800s. And he figured out that a nucleic acid is a molecule made of a sugar, a phosphate, and a nitrogenous base, right? The nitrogenous base being all the A's, T's, C's, and G's, right? And so these things joined together made one monomer of a DNA polymer, right? These are words we've covered, things that are in the back of our heads, right? So it's a sugar, a phosphate, and then some base, either adenine, thymine, cytosine, or guanine. Okie dokie. You will not need to draw DNA molecules on the exam, but just so we kind of understand what's going on, right? It looks like this, right? Here's our 5-carbon sugar. Here's our phosphate group, right? We've seen phosphate groups and sugar rings before, that's fine. And then here's that nitrogenous base, right? It's a base as opposed to being an acid, and it's got nitrogen in it. Cool, fair, makes sense, right? And so these bases come in one of 2, we kind of split them into 2 groups basically. So we have these purines, which are made of 2 rings, which contain nitrogen, right? And that would be adenine and guanine, right? You probably, if you're in the lab, right, you made rings out of little ball and stick models. We kind of have a vague idea of what this is. And then we have the purines that are made of 2 rings and the pyrimidines that are made of a single 6-sided ring, right? So it's a carbon ring with some nitrogen in it. And so in the pyrimidines, we have cytosine, thymine, and then uracil that's only going to be an RNA. That will come later. We'll talk more about RNA later. But when DNA is converted to RNA, thymine is swapped out for uracil. All your T's turn to U's, basically, in your genetic code. But all right, so we've got these 2-ring purines, 1-ring pyrimidines, And so every little monomer in DNA is going to have one of those stuck on out here, right? Attached to its phosphate, or attached to its sugar, which is attached to a phosphate. All right. So we're going to say 5' and 3' a lot. It takes a while to kind of like wrap your head around it. You may want to like go home and draw pictures or kind of think about this a bit, because we're going to talk about 5 to 3 and 3 to 5, and it's going to actually kind of matter for better or worse. So I'll keep saying it and hopefully it'll kind of sort itself over time. So, right, we've got these monomers, phosphate sugar, nitrogen base. We're going to attach it to another phosphate sugar, nitrogenous base. Cool. How do we do that? We're going to attach the phosphate on this guy to the little hydroxyl, the little OH on this guy. So another way to kind of talk this through, if you remember, I don't know if Eagle would do this, Menzel would probably do this. To kind of orient ourselves in these molecules, we give the carbons numbers, just to kind of name them and give us a point of reference. So there's a carbon here, 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 and here. And so we number them, 1, 2, 3, 4, 5. Right, since this is carbon 5, I'm going to call this side of the molecule the 5' end. Right, this is my third carbon, so it's 3'. Right, same thing here. This carbon here, same carbon. This is the 3' carbon. So what I'm doing is attaching my 5' phosphate to my 3' hydroxyl. Yeah? Yeah. So the phosphate is stuck to carbon number 5, right? So here's carbon number 5, here's its little phosphate group. This is carbon 3. It had a little hydroxyl group, and we're going to attach the phosphate. It's going to make a little phosphodiester bond between carbon 5 of this guy and carbon 3 of that guy. So there is a bond between the 5' phosphate of a nucleotide and the 3' hydroxyl, right, OH of the next nucleotide, which means, and it makes sense, right, because DNA carries information, we're going to read it. So if you think about like I read from left to right, right, DNA is also going to have a direction. There's a direction you're supposed to read it. So it's going to get put together in a direction from 5' to 3'. Right, so when your cells or your enzymes and things are reading DNA, right, they're reading them in a direction, like their own version of left to right, from the 5' end of the molecule to the 3' end. Okay. Okay. So we know what the pieces are, but what does the whole big polymer look like? Like, is it just a loose chain of bases and phosphates and stuff just kind of sitting like a big pile of spaghetti? Maybe. I don't know. No, it's not. So Watson and Crick are given credit for determining the actual kind of structure and shape of a DNA molecule. But if we're honest and we kind of pay attention, they actually did no scientific experiments whatsoever. They kind of took results of other people and kind of put them together in their model of what DNA looks like. So let's back up. Whose stuff were they using? So Chargaff, Erwin Chargaff is a fun name. So he found, looking at DNA molecules from different organisms, that the nucleotide composition, right, the numbers of A's, T's, C's, and G's, was different depending on who you took the molecule from, right, which makes sense, right? My genome is different than your genome. Our genomes are different than a tree genome, right? So they're going to have different A's, T's, C's, and G's. I mean, at the base of it, that's how it works. He didn't know, but we know. But the ratios of bases, A's and T's and C's and G's, followed certain rules called Chargaff's rules. Cool. So rule 1, the amount of adenine in your DNA is equal to the amount of thymine. So your number of A's is equal to the number of T's in your DNA. Likewise, the amount of cytosine in your DNA is equal to the amount of guanine. So you have the same number of C's as you have G's in your DNA. Rule 2 is that these ratios, right, the number of C's and G's I have and the number of A's and T's I have will differ between me and anybody else's DNA, right? And of course, the order that they go in, right? So I will always have the same number of C's and G's. I will always have the same number of A's and T's, but the numbers in my DNA will be different from anybody else's. especially if they're of other species. Yeah, okay. So Chargaff figured that out. Rosalind Franklin, if we're all very honest with ourselves, actually figured out the shape of DNA before Watson and Crick. So she was doing X-ray diffraction, which is just kind of this way to image molecules. And so she actually had pictures of DNA, right? And it's kind of hard to see in this image, but right, she had pictures of, right, DNA forming a helix. She knew what size and what shape the helix was. She discovered that DNA is helical. Not like you'd have to come up with her name for a fill in the blank. She may show up like as part of a question, but you don't have to like have her memorized specifically, I would say. All right. And so Watson and Crick learned of Rosalind's results before she was able to publish them, and kind of borrowed them a bit. Not the coolest move. And so they took Chargaff's results, Franklin's results, and some other people's work as well, and deduced the actual kind of full molecular, this is their like plastic molecular model of DNA. They performed 0 experiments to do this. They just kind of pulled everybody else's stuff, made their DNA model, and then won the Nobel Prize, along with Wilkins, this other guy who never shows up in the pictures for some reason. Right. So sometimes science isn't fair. It's true, it happens. Data miners still exist. But yeah, but Watson and Crick are usually kind of accredited with the discovery of DNA structure. Rosalind Franklin, we kind of all also know, actually discovered it, but that's fine. I mean, it's not fine, but it is. So let's talk about this DNA structure that everybody was working so hard to figure out. DNA, it's a double helix, right? We have all seen pictures of DNA on stuff. People use it to illustrate, you know, it just gets put on things like weird little logos, right? And it's made of 2 strands that are wound around each other, right? The DNA double helix. But let's look at one strand first and kind of get an idea of what we're looking at here. So each strand, right, of that double helix is composed of, right, these linkages that we just talked about, right? 3' carbon attached to a phosphate to a 5', right? So going from 5' to 3', right? It's sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down, the whole long thing. The bases aren't involved in this at all, right? The bases are not involved in the polymerization of DNA, as it were. They're just all these bases, right? All these purines and pyrimidines are just hanging off in the middle for now. So this phosphate, this phosphodiester backbone is formed, and it forms, again, from 5' to 3', right? From the 5 carbon to the 3 carbon, right? It's kind of the reading direction of DNA, right? So each of the strands is composed of this phosphodiester backbone. So we can have 2 separate strands. Okay, so those 2 strands then, right? These blue ribbons then represent that phosphodiester backbone, right? They're just phosphates and sugars, right? All wound around each other. The bases are kind of pointed internally, right? So this is where you line up your A's and T's and your C's and G's, and that's actually what connects the 2 DNA strands together, right? Into this double helix shape, right? And they're held together by hydrogen bonds. Hydrogen bonds never go away, right? So we've got more hydrogen bonding happening. So it always happens, again, in these pairs, right? So guanine will always bond with cytosine, and it will always have 3 hydrogen bonds between them. Adenine will always bind to thymine with 2 hydrogen bonds between them. And it makes this nice, even kind of balance, right? The double helix isn't this weird, like wonky shape, right? It's a very even, right? The diameter is the same all the way down, kind of tidy molecule, right? This results in us having 2 specific base pairs, right? As you go down the helix, you either have a C and a G or an A and a T, always, right? So we've got this consistent diameter double helix molecule. Okay, that's cool, fine. The result is, if you look at a DNA strand, that you end in end up with two complementary DNA strands, right? Because if we think of A's and T's and C's and G's, it's like the two strands are inverses of each other, right? Everywhere there's a C on this side, there's a G on this side. Everywhere there's an A on this side, there's a T on this side, and vice versa. So we call the strands anti-parallel, because the other thing that happens, more 5 and 3 problems, right? So we've got this strand of DNA. Here's our 5' end, right? All these bases get added 5 to 3'. Its little partner strand here is going the other direction, right? So 5' on this strand is here. The 5' end on this strand is here, right? You can see these little, the sugars are facing opposite directions. So the DNA code is running opposite directions. So they are anti-parallel. So they're parallel, but kind of back to front, you could say, right? So we have one strand going 5' to 3', and the complementary strand then is 3' to 5'. Okay, okay. All right. So we know what DNA looks like. Theoretically, we can make a copy of DNA now, right? Sure, sure. So we're going to talk about how DNA gets replicated, and then we're going to talk about what is required to get DNA to make a copy of itself, basically. Right? Because that's the whole thing. Meiosis is all based on the fact that we can get through S phase and make copies of everything. So how does that happen? There are kind of a whole lot of moving parts in DNA replication. As you can imagine, it is like the most important process in your body. If your body cannot replicate DNA, everything will fall to bits, right? You need to make copies of your DNA, and they need to be good copies, right? So accurately copying your DNA is critical, right, to making new cells via mitosis, making gametes via meiosis, either way. And so when Watson and Crick kind of proposed this double helix model, they figured that when DNA is copied, right, I've got these complementary strands. So theoretically, I can look at one strand and make a copy, right? You know what matches up, A's and T's and C's and G's. You can make a copy that way. So one strand could determine its copy strand. Cool. So these parental strands, right? I have my chromosome, these two little strands wound together. To go through meiosis, I need to make a copy of it, right? So we call that a parent strand and a daughter strand, sure. Right? So one parental helix, my two strands wound together, need to go through S phase and become two daughter double-stranded pieces of DNA. So I need to go from 2 strands to 4 strands. And then those daughter strands can be divided up during cell division later on as necessary. All right, cool. So we're not going to walk through kind of the whole process of how we got to how DNA is copied. I usually do, but we're not going to. We're just going to skip ahead. So there were lots of hypotheses about how DNA could be copied, like what the strands were actually doing to make copies of themselves. The correct answer was the semi-conservative model. So the semi-conservative model states that daughter strands, right? So here's our 2 daughter strands in orange. Here's our blue parental strands. So when you make these 2 kind of daughter chromosomes, right, we go through S phase and we end up with 2 copies. Each of them is made of 1 parental strand and 1 copy, right? 1 parental strand and 1 copy, okay? So it's called the semi-conservative model of DNA replication, right? So what we're going to see is basically what's going to happen because these 2 strands are complementary. We can pull them apart, make a copy of each of them, and end up with 2 DNA strands that look exactly like the one we started with. Okie dokie. So what do you need to do DNA replication? Just 3 things, right? That sounds simple enough, right? So we need something to copy. Duh. You need a DNA strand to make a copy of, starters. You need something to do the copying. This is going to be kind of a bunch of enzymes, but that's what enzymes do, right? They do work, they do things in the cell. So we're going to have enzymes that are going to be making the copy, and then you need something for the enzymes to make the copy out of. So those are nucleotide triphosphates, right? Because if I'm making a copy of this strand of DNA, I need a pile of A's, T's, C's, and G's over here to make the copy out of, right? So I need a thing to copy, a DNA strand. I need something to do the copying, a bunch of enzymes. And then I need stuff to make the copy out of, a bunch of nucleotides. Cool. when you, just random, when you are amplifying DNA, if you're doing DNA sequencing and you want to make a whole bunch of copies of DNA, you literally put the DNA you extracted, a bunch of enzymes, and a bunch of nucleotides in a tube and heat it up, and you make DNA. That's all it takes. It's pretty cool. Anyway, so it's just, there's this little machine. You're just like, boop, make me some DNA. And it's like, and it does it. All right. That's what the machine actually says it. No, it doesn't. So stages of DNA replication. Again, kind of intuitive. You start, you copy, and then you stop. But as with kind of all things science, we give them specific names, right? So starting is initiation. So this is where replication begins. And it's going to require some specific parts to kind of get the copying process started. So we have initiation. Elongation is that process of the enzyme, DNA polymerase, spoilers, right? DNA polymerase adding new bases to the copy of the DNA. So we're going to elongate our new strand of DNA. And then when we're done, we're going to terminate the replication process. All right, so we're going to start, we're going to make a copy, and then we're going to stop. Easy peasy. Sort of. So DNA polymerase is going to be kind of our go-to enzyme for the bulk of this process. And there are different kinds of DNA polymerases, we'll get there. But DNA polymerase, what it does, right, it can look at our, what we're going to call our template strand, right? This is the strand of DNA I'm making a copy of. It can look at this base, right? Oh, there's a C here. I need to attach a G. There's an A here. I need to attach an A. And so it just works down the strand, pulling in the complementary bases and attaching them on. So that is what DNA polymerase does. Matches existing DNA bases with the complementary nucleotides, right? That would make that complementary strand. Right? Specifically, these are little triphosphates, so they kind of work like ATP. So they come in here and they attach to this hydroxyl, right? And we're going to pop these phosphates off to attach a new nucleotide to it, right? We create a new phosphodiester backbone with complementary nucleotides in the middle. All right, boop. DNA polymerases all have a few things in common. We're coming back to this 5' 3' situation. All right, so again, we're moving from 5' to 3'. So all DNA polymerases are going to add their bases to the 3' end, right? Because if I'm starting 5', I'm adding bases on the 3' end, because the 5' is over here, 3 is over here. I'm just going to keep adding them on, right? So they all move from 5' to 3', and they require a primer, right? You need a little piece to get you started. So there's going to be another enzyme, we'll get to him later, and he's going to be like, here's a primer, you need to start copying here, this little guy. He's like, I'm putting this primer here. Polymerase, you need to start at the primer and start making a copy. And then polymerase does its thing. Yeah. So all polymerases add to the 3' end, moving from 5' to 3', and they got to have a primer. All right. So we're going to walk through DNA replication in prokaryotes because it's easier as things go. Eukaryotes, as always, are a bit more complicated. So we're talking about polymerases. We're going to talk about discontinuous polymerization. And then we're going to talk about the replication fork, most of which will make sense once we see a picture of it. So simple stuff. Prokaryotes are easy, right? We've talked about prokaryotes. It's just got like the one little circle chromosome in the middle. So all we got to do is make a copy of this one little chromosome. Easy enough. And we talked about this when we went through binary fission, right? You've got this circular chromosome, and it kind of has like a start spot and an end spot, right? It goes from the origin of replication, ends at the terminus. So we're going to start copying at the origin, copy down to the terminus, and then have 2 copies of our DNA. Easy enough. This unit, right, a piece of DNA that has an origin is called a replicon, right? So in prokaryotes, you basically have a single replicon. I've got this one chromosome. It's got one start point. It's one replicon of DNA. Okie dokie. Let's see. Right. So this is basically what the entire process looks like. We're going to pick it apart a little bit more, but right, we've got a start spot and an end spot. We're going to attach all of our primers and enzymes over at the start spot and start making a copy, right? So one of these guys is making a copy of one side of the DNA. one of these guys is making a copy of the other side of the DNA. So they're going to work all the way around till they get to this termination spot, right? We're going to pull these two pieces apart and end up with two copies of our DNA. Easy peasy-ish. Easy enough, right? So that gets us to words. We're going to have to pick up a lot of vocab. Talk about, all right, I'll give you exo and endonucleases real quick and then we'll be done. So ACEs, right? These are enzymes that are doing something with nucleotides, right? Nucleases. Exo means outside. Exonucleases can cut nucleotides off the ends of DNA. Endo means inside, can cut nucleotides out of the middle of DNA. All right. And so we will pick up then at polymerases on Friday.